Welcome to the tutorial video for Catnsap. This is an online tool designed for searching for conserved alternative proteins in plants and animals. These proteins can arise from various types of alternative splicing, alternative transcription start sites, and alternative cleavage and polyadenylation. Catnsap searches for instances where the amino acid sequence of one isoform conservatively changes relative to the other isoform. Thus, the user needs to provide a pair of proteins. Catnsap can be used with NCBI protein accession numbers or sequences. To find the accession numbers of your protein pair, go to NCBI and find the gene that produces the protein pair of interest. For example, I'll type Arabidopsis repto7. Here I select gene. Here is our gene. Go to the gene diagram. Unfold the gene structure by left-click on the green gene diagram. Purple diagrams correspond to mRNAs and red diagrams correspond to proteins. I am interested in this event where an exon is skipped. I need two isoforms which only differ in the presence of this alternative exon. Here they are. To copy their accession numbers, hover your mouse over the corresponding isoform. This is the needed accession number, so copy it and paste it to the field labeled NCBI accession mode. Copy the second isoform accession number and paste it into the same field after a comma. Now we are ready to submit our query. Catnsap will look for similar protein pairs in its database of alternative protein sequences extracted from RefSeq and GenBank. The full database contains alternative proteins from 176 plant and 701 animal species. To speed up the search, we have created a reduced database that contains 176 plants and 100 animals. Press Submit. Catnsap will blast the query sequences against its database of proteins. It will then create preliminary orthologous pairs and carefully evaluate their similarity to the query pair, using a machine learning model. The similarity of identified pairs will be expressed as similarity scores, where a score of 1 indicates complete identity. Now we are on the output page. It displays identified pairs of isoforms from different species, sorted from the most to least similar. If we align the sequences, we can clearly observe the differences in the alternatively spliced regions of the proteins. Press Align All to start the alignment. Let's find the region undergoing alternative splicing. Here it is. The identified orthologous isoforms differ in the presence of this short amino acid sequence. As we move closer to the end of the list, the conservation of this region becomes questionable and eventually disappears, as reflected by a very low similarity score of 0.0. You can delete unwanted sequences by pressing Delete on your keyboard and realign the results by clicking Align All again. You can also save the displayed alignment by clicking Save Alignment. We can look at the exon-intron structures of the aligned isoforms. To get a clearer picture, let's build the structures for 30 isoforms first. Set the number of sequences here, then press Align 30 sequences. Then press Structure. Here is our alternative exon, just like it is depicted on the gene diagram on NCBI. Orthologous exons are aligned one below the other. Interestingly, we can see that in Arabidopsis thaliana, exons 7 and 8 have merged into one. If a species has gained an insertion within an exon, it will appear as a wide gap in the other species. The more species are aligned for the structure view, the more chaotic this page will become. Let's add more sequences. First, you need to press Restore All Sequences, then enter the desired number and align them. As we now have more divergent sequences in the alignment, the number and size of wide gaps have increased in the less conserved regions of the proteins. If you press Structure when the sequences haven't been aligned, Catnsap will build structures for the number of sequences entered here. Very long introns can make it difficult to view the regions affected by alternative splicing. To shorten the length of introns, click the Compressed Introns button. Please note that Catnsap only builds structures for the coding regions and is not aware of the untranslated regions. Orthologous pairs identified by Catnsap can be downloaded by clicking Download Sequences. The archive folder contains two types of files. Best hits in a species contains the same sequences as the ones shown on the output page. All hits in a species also includes pairs from paralogous genes or other alternative splicing events in the gene. Sometimes the actual best hit is not the one with the highest similarity score, so all identified pairs in a species are provided. The contents of the files can be easily pasted into your alignment tool. Thank you for watching this video. More details are available on the help page.